Good morning, Jai Jalaram. Semester 3, paper 301, Microbial Diversity. We are going to study about the identification of gram-negative bacteria on the basis of biochemical characterization. As earlier we have understood that what is the meaning of biochemical reactions. The reactions which are occurring in the chemical manner in the biological system, these reactions are known as biochemical reactions, and these reactions are carried out by particular enzymes. As we know, every enzyme is working and functioning with a particular substrate, and there will be production of a particular product. Depending upon this product, if we can identify, if we can detect, if we can observe this product, then we can say that the substrate has been utilized with the help of particular enzymes. Enzymes are working with substrate just like lock and key arrangement, so they are very specific. Let us see how carbohydrate substrates which are helpful to us for the identification. Starch being the polysaccharide, we are using starch agar plate. As we know that starch with iodine will give us blue color. If organisms have produced amylase enzyme, it means they are going to utilize the starch. That means the blue color in presence of iodine will not be produced, or there will be clear zone around the colony. If there is clear zone around the colony, on the next day when we will add the reagent iodine, then those colonies will be labeled as amylase producing colonies. This is the thing which we are going to see in the starch agar plate. Next is the sugar utilization. With the help of 6 common sugar, as we have seen in the slide, it will be some of them are monosaccharides, some of them are disaccharide, and one alcoholic sugar also we are going to take. So glucose, lactose, maltose, sucrose and mannitol as an alcoholic sugar, and xylose as a pentose. So we are going to have representative sugars from the family of carbohydrate, sugar, and these are going to have certain enzymatic reactions. So at the end of that there will be production of acid and gas. Certain indicator dyes are added in this medium so that this dye will convert its color from the alkaline state into the acidic state. The color will be changed, as here in case of bromocresol purple, the bromocresol purple will turn yellow, and if there is gas production, Durham's tube will show bubbles and it indicates the gas production that was connected with 6 common sugar utilization. Glucose phosphate broth is taken for the purpose of methyl red test and for the purpose of Voges-Proskauer test. Citrate slant will be taken for the purpose to find out whether citrate is utilized by these organisms or not, and triple sugar iron agar slant is also checked for the presence of certain bacteria. Let us see. In this starch agar plate, we are going to see the particular organisms if they have produced clear zone around the colony. The blue color indicates the presence of starch and the colorless area indicates the absence of starch with the iodine as an indicator. In case of 6 common sugar, we can say these 6 common sugar are seen as in this particular tube it is bromocresol purple with Durham tube, and next day if organisms will utilize this sugar they will be producing yellow color because bromocresol purple will turn to yellow and gas will be produced in this Durham's tube. So this is the reaction in 6 common sugar tubes. Next is glucose phosphate broth. In this glucose phosphate group, when we are going to check methyl red, we are going to have different pH strips. We are going to have different pH strip, which will tell us that what is the pH of the particular medium. This pH strip, when we will dip it and we can check, methyl red test will indicate what is the pH of the medium, and Voges-Proskes test, in which we are going to add alpha naphthol and 40% KOH for the production of acetyl methyl carbinol. There will be coloration which will be pink color. So in this VP test, if the pink color will develop, we can say VP test is positive. Afterwards, we are having citrate slant. In this kind of the citrate slant, we are going to check whether next day the color of the slant changes from this greenish into the bluish color, which will indicate the production of citrate. And next is TSI slant. In this TSI slant, we are going to check triple sugar iron, that means 3 sugars are utilized. There is also ferric salt are added here, which will turn this slant into blackening. So there will be 3 reactions which we are going to check in this TSI slant, that we will see in the description. So this was all about the carbohydrate media which we are going to use for the identification on the basis of carbohydrate utilizing enzymes. In the next video, we will see proteinic substrates to check proteolytic enzyme ability of the given colony culture.